Welcome everybody to the Parkinson's portion of our Neurological Symposium, where we're proud to partner with MGB, or Mass General and Brigham and Women's Hospital. And this event, I guess probably one of the good things that came out of COVID is we started broadcasting this and offering it worldwide. So just to let everybody know that there is 5 countries, 16 states, and 137 towns and cities that have signed up and are tuning in, so welcome. I would like to thank, first and foremost, these very, very talented physicians for their time, donating their time. But also like to thank our major sponsor. You can see many of the biotech pharma companies have supported this, and it helps us offer this for free. But in particular, first and foremost, Leader Bank, and then our 2 major sponsor, InfraCare RX and Novartis. So thank you all. I hope you all enjoy the information that's going to be shared. It's a lot of exciting information, and we're just proud to be able to offer this and being part of what our foundation delivers to the neurological community. Thanks so much.
Thanks, Brian, and to the McCourt family and foundation for bringing us together for this worthy endeavor. We're focusing in this hour and a half on Parkinson's disease. Do I just signal? Thank you, Miguel. I'll be sharing with you some of our enthusiasm for a rather novel aspiration in Parkinson's and neurodegeneration more broadly about moving earlier in the disease process to actually have a chance to prevent the disease or delay its onset, by telling you about some advances for the prospects of preventing Parkinson's disease and related disorders. My name is Michael Schwarzschild. I'm a movement disorders neurologist at Massachusetts General Hospital, part of Mass General Brigham, and I'll be joined by my colleagues from the Brigham for the follow-on after this 10 or 15 minutes to go over what's been rapidly evolving to make prospects for preventing Parkinson's disease for the first time realistic. First, I'll go over why it is prevention, if it isn't obvious, is such a good idea to consider. And then I'll go over some of the advances that have informed us about who's at high risk, how to measure progression or prevention, and what to try in the first trials that are just beginning now. And if it isn't already obvious, this is a partnership between the investigators and all the stakeholders, but most importantly, people with Parkinson's, and by with Parkinson's, I mean in this broader framework, people who are at high risk, for whom we now have prospects of making it less likely that it'll manifest in the first place. So why Parkinson's? Why prevention? The principle actually is nicely captured here in an ounce of prevention is worth a pound of cure, and interestingly further, that's from a quote from Ben Franklin from almost 300 years ago, when he was talking about a framework for developing preventative therapies. In his case, he was focused on fire prevention in Philadelphia, but it's a very nice framework, and I'll apply it to how one thinks about it in medicine, and certainly Parkinson's as well, that prevention can be applied at multiple stages of a disease developing or playing out. Medically, there's a definition that it's action taken to decrease the chance of getting a disease or condition. But in staging it out, when we think of disease modification for Parkinson's disease, traditionally it's only been after Parkinson's has been diagnosed, where we would call that tertiary prevention of trying to prevent disease from getting worse or preventing complications like cognitive decline or falls and the like. but and this is just to cite Franklin's paper on protection of towns from fire. Right, so tertiary prevention, in his view, would have been after the fire starts, preventing it from burning down the next building. And there he was talking about tools, interventions like fire engines. He actually coveted the Boston Fire Department and shouted it out as something that Philadelphia should aim for, ladders and the like. But moving earlier, that when there's this idea of secondary prevention when the process has already started, but it hasn't spread to become a problem. And there you can think of it as the embers in bedpans or sheet warmers or in fireplaces that he focused on as well, where preventing that from becoming a fire is a way of thinking of secondary prevention. And then primary prevention is before there's any fire in the first place, preventing the conditions in which the fire can take place, also very applicable to Parkinson's and other neurodegenerative diseases. And there, of course, and this is what he spent most of the time on, was on chimney sweeps and the soot that builds up that can create that. And that accretion, which he focused on, is just sort of foreshadowing, as it turns out, for one of the pathological precursors for Parkinson's disease that we now are framing our own understanding of the disease by something called alpha-synuclein that you'll hear more about in a precision medicine approaches that will be discussed. And clearing out that alpha-synuclein is a strategy that harkens back to Franklin's framing way back when. So good idea to prevent things. What are the precedents for prevention in Parkinson's disease? And I kind of broke them down into two kinds. Ones that are unfortunate precedents, things where we're forced towards prevention because our approach to trying to actually slow down the disease hasn't gone so well. But there's also some recent encouraging precedents for prevention within neurodegeneration and outside that I'll just touch on. So here's what looks to be harsh in this cradle-to-grave metaphor from my colleague Alberto Espe, who's highlighted that these great ideas, as they're born and then developed and eventually get tested, and I should say, if it isn't obvious, the framework for a lot of the research that we're pursuing is the actual experiments, if you will, which in humans we call clinical trials when they're done well. And it's nurturing these candidate ideas that have been pursued from the cradle to, unfortunately, in all cases so far, results of trials that haven't demonstrated that the candidate disease-modifying therapy has actually slowed down the disease. This is an older slide, and since I've been using it, I've only been adding tombstones. Most recently, there's the Ozempic-type drug, a major trial for one form that was disappointing in not demonstrating slowing down the disease for a drug called exenatide. So this looks very harsh, and this is the negative precedent that's stimulated us to look towards prevention to some extent. But I don't want to overstate the graveyard theme here, because while it might suggest that the definition of insanity is doing the same thing over and over again and expecting different results, which is what people have sometimes criticized our investment over decades in these kinds of trials, with nothing turning out to slow down the disease, at least in Parkinson's so far. But if you look closer, it's actually quite different. I don't think that adage applies, because we actually are learning an awful lot about how to do these trials better and how to select better therapeutics. So I remain hopeful, and just as examples, that there are some very nurturing cradles that are out there that are targets that are in phase 2 or phase 3, medium and more advanced trials that some of you may hear about, we can talk about, that are encouraging for slowing down the disease in people who have the disease already. And this is just to point out that in addition to medications, there's also strategies that are being pursued in clinical trials that relate to risk factors, and I'm just going to highlight one that's in a fairly advanced NIH-supported trial called SPARKS-3, that's aerobic exercise, randomized, rigorously done trial that we should be learning the results over the next couple of years. So there are still many hopeful approaches to slowing down the disease that still have cradles that are being pursued. But the harshness of the results so far combined with opportunities arising, has led us to look earlier in the disease and again, to play on an adage, closing the barn door after the horse is bolted is something that can apply to when we apply these trials. And if we are testing therapeutics that can slow down the disease after the disease is already running, there may be better opportunities if we can go earlier while the horse is still in the barn, if you will. So in that sense, the negative results that we've had have been tempered by the fact that we're learning a lot from these trials, and there's still some very promising therapeutics for people who have Parkinson's that are being tested for the prospect of slowing it down. But for preventing it or delaying it, there's also some other precedents I just wanted to touch on. One is actually the first approved drug, sort of a watershed moment just a couple years ago for any disease that the FDA has approved for preventing or delaying the onset of a disease, which is applied to young people who are at risk for but do not yet have diabetes, for whom there's now an approved treatment that makes that less likely. And then precedent also, and I know there was just a session on Alzheimer's disease where from other neurodegenerative diseases that are linked, to be sure, to Parkinson's disease, like the HD stands for Huntington's disease, the AD for Alzheimer's, and PD for Parkinson's. There's a framework, and as using the same prevention trial design puzzle, piecing together of who, what, and how to try to target for designing these trials, we can learn from trials that are ongoing. There's been no prevention demonstrated so far in these other diseases either, but they're further along in identifying, for example, who to try to prevent Huntington's disease. And of course, it's a largely genetic disease, so people who are genetically at risk, that's an opportunity that Alzheimer's and Parkinson's has to a much lesser extent. And in Alzheimer's disease, there are biomarkers that with brain scans, spinal fluid, now blood tests that are fairly reliable for not only understanding who's at increased risk for the disease, but also for measuring how it progresses. And so that sets up a roadmap for Parkinson's disease, and this is just one last slide. It's a little bit thick here, but it illustrates that primary, secondary, tertiary prevention concept applied to Alzheimer's disease, where in this life cycle on the x-axis, where people have no Alzheimer's pathology, then go on to have the pathology that can now be measured in the blood or in brain scans, but people don't yet have cognitive decline or dementia, to the later stages when now the process is leading to clinical symptoms. Previous trials have only focused on so-called tertiary prevention, but now there are a number of trials focused on secondary and even primary prevention. So the rest here I'd like to just touch on are not only why this is something that's so compelling to pursue for Parkinson's, trying to prevent it in the first place, but why it's now an opportunity because of for the prospect that it's actually a realistic kind of trial to pursue, and that's because of advances that have been made in these other interrogative categories. Probably most notably over the recent years and decade or so is in who is at particularly high risk of disease. Because unlike Huntington's disease, fortunately perhaps for most families, that Parkinson's doesn't run strong in families. It certainly can run strong enough in some, so that's an at-risk factor of people who are genetically at risk, but there are other factors as well. And I'm just going to touch on these biomarkers like we're seeing with Alzheimer's that you'll hear more about, and this prodromal period that precedes the time someone is diagnosed with Parkinson's disease, and sometimes even before there are actually symptoms of the disease. And this was just to highlight that just like McCourt Foundation's investment in education, we've been early on very excited by this possibility. We've been bringing people together, stakeholders from around the world, on this emerging possibility of trying to prevent Parkinson's disease. And at our first conference in 2021, then a couple of years back in 2022 with stakeholders from FDA and NIH and industry and various foundations and philanthropies supporting this. And then most recently, just last year on the Harbor. And we're trying to launch these first trials that are now taking place in Europe, in the US, and in China. And again, it's because of our advances in understanding who's at such high risk. So this last sort of schematic here I have is just a depiction of how there's been a shift in the last decade or so, where we've been studying really only tertiary prevention, if that framework has stuck, in Parkinson's disease. Sort of classical clinical Parkinson's is people who have received a diagnosis because their symptoms have progressed, usually over a few years, to the point where they saw a doctor and got a clinical diagnosis. And it's under that street lamp, if you will, where we've done all our studying, all our clinical trials. And that's depicted in this, again, this sort of lifetime framework where that period starts when there's a traditional diagnosis until one finishes their time on Earth. And during that period, there's those lines that are angling up towards 100% are the involvement of the green one, how the shaking or slowness gets worse and worse over time till it's maximal at the end. But then, as many of you know, there's non-motor features that actually often precede the onset of the motor features, starting years before, but they also increase. And then, of course, the underlying pathophysiology, abnormalities in the cells and the molecules, that starts even earlier. And so all our trials, though, as you might notice, in that last period of basically when one lives with the diagnosed disease, we've always tried to do the trials which are depicted by little circles, the red ones being more advanced phase 3 trials, the blue ones earlier trials, where they're all kind of pushed up against the earliest diagnosis opportunity, because again, doing things early, as close as you can before the horse gets out of the barn, has been a principle that's been adhered to. This last slide here with schematic just shows that another street lamp has been turned on, if you will, and it's for that period that precedes a diagnosis, before you see a neurologist or a movement disorders neurologist, when these processes that I described are all happening. Obviously, most proximate is whatever that was slowing one down to the point where one led to a diagnosis was also present more subtly, whether it was a tremor or slowness or imbalance for years before. So it's called the motor, the clinical early motor phase, prodromal phase, and then prior to that, a non-motor phase. And I'll just spend a minute on that. Prior to that, actually, and you'll hear more about this in a few minutes, is a preclinical, that is no manifestations by symptoms or clinical signs at all, but the process may have already started, and there's evidence of that by biomarkers like those that measure synuclein that have really been a breakthrough in just the last few years. I just wanted to mention, those living with Parkinson's generally have known this before clinicians and scientists knew it decades ago, that there are telltale clinical signs that precede by years and sometimes by decades the disease that have, in some cases, are very predictive and specific. One that is subtle, but strongly associated with the disease is a loss of sense of smell. That is not entirely specific, because you can have that from head trauma, COVID, other things. So just losing one's sense of smell doesn't mean one will get Parkinson's. But the vast majority of people who develop Parkinson's have lost substantial portion of their sense of smell years before. And then the last symptom that I wanted to just share, if it's new to you, but to also highlight how it's helping us find who's at high risk for enrolling into prevention trials, is something called REM sleep behavior disorder, essentially acting out one's dreams. And it's something that a bed partner usually notices, because one is sleeping when they're actually manifesting that, typically by yelling or thrashing in one's sleep. It's most definitively assessed by a sleep study that is done clinically. But I wanted to, as the last depiction of this, share the first real public figure who so
Alan
nicely
Alda is with us this morning for an announcement that he
talked
wants to
about
make.
this.
So good to have you here.
Thank you. It's great to be here.
How are you doing?
I'm doing great. You might be surprised to hear that. When I haven't said in public until now that I've been diagnosed with Parkinson's disease. And the reason that I want to talk about it in public is that I was diagnosed
Sorry, Miguel, there's a part, maybe you can just play, maybe it'll come up soon, the part where he talks about the sleep disorder, but go ahead and play it, and I don't know if this will jump to it, but So this is Alan Alda, who actually,
I was diagnosed 3.5 years ago, and I've had a full life since then. I've acted, I've given talks, I help at the
there's
Older
a
Center
second
for
marker
Communicating
that
Stress a couple of weeks
jumps to
talking
where he
about
talks about
the
REM
podcast,
sleep behavior
and I
disorder.
could see my thumb twitch in some
here-
dots, and I thought, it's probably only a matter of time before somebody does a story about this
Right.
from a sad,
What was that diagnosis like when you got it?
well, I asked for a scan because I thought I might have it. I read an article by Jane Brody in the
That's
New York Times that indicated that if you have,
Oh, that's good. That was it. Yeah, so Alan Alda also has an institute for scientific communication. And he's living it here and sharing his diagnosis and also the insight of what preceded it with his REM sleep behavior disorder. So that you were just at the exact right point just then.
and the families can suffer, but in the very beginning, to be immobilized by fear and think the worst thing has happened to you, it hasn't. Suffering with really severe symptoms, you know.
It was the
symptoms can
point
get
before.
very bad, and and the thing I want folks to know, and so months later, a little twitch in my
A little before
thumb,
that,
you know.
it's
What
the
other
second marker
symptoms did
that's,
you start
that's
to
written.
notice?
I didn't have any symptoms. The doctor said, why do you want to scan? You don't have
Good
any
enough.
symptoms. And I said, I want to know if there's anything I can do. I want to
Oh.
do it before things start to show up. And so months later, a little twitch in my thumb.
Sorry, it's 10 seconds before, but I'm not
And
sure it's worth trying to go
thing I
back to
want
it.
Folks to know.
It's where the
and this
second
is
marker
not
is in
to
the,
shortchange people who are any symptoms. The doctors, by acting out your dreams, I mean, I
A little
was
before.
having a dream that someone was attacking me and I threw a sack of
backwards,
potatoes at
earlier.
a symptom that where nothing else showed, that indicated that if you have, if you act out your dreams, there's a good chance that might be
That's-
a very early symptom that where nothing else shows. And by acting out your dreams, I mean, I was having a dream that someone was attacking me and I threw a sack of potatoes at them. But what I was really doing is throwing a pillow at my wife.
Wow.
Wow.
Yeah.
So what other symptoms did you start to notice?
I didn't have any symptoms. The
Nothing.
doctor said, why do you want to scan? You don't have any
Wow.
symptoms. And I said, I want to know if there's anything I can do. I want to do it before things start to show
That's
up.
great. That's great. So Alan Alda, who many of us grew up with, and as I was saying, actually has an institute for scientific communication, is living his later chapter work here, not only in the context of the Parkinson's that he's developed, but also in educating people about this clue that was missed by so many, that as it played out in his case, where he learned about the disease and wanted to prevent it when he learned that he had something that could precede it earlier. And that's what REM sleep behavior disorder is, and that's very much what we're pursuing here. So I think this conference, which we're actually holding again, it's now every 2 years, is next May, which is pulling in family members of people with Parkinson's who are at risk or otherwise at risk, as well as the broader research community. And it precedes the World Parkinson Congress in May, also in Phoenix, which is something that's even more designed to pull in the patient along with the Parkinson's, living with Parkinson's community, as well as scientists. And so these are wonderful opportunities, these conferences, to get more involved. And you can follow up if you have any interest there. And I'm just going to turn next to my colleagues. I think Vic is going to tell us more about precision medicine. Thank you.
Thank you so much. Michael has given a great introduction to Parkinson's disease. I'm going to pivot now. I lead the Movement Disorders Division at Brigham and Women's Hospital in Boston. I'm a physician scientist. I'm going to be speaking with one of my colleagues here today, Rich Krolewski. Both of us are very interested in developing the next generation of treatments for Parkinson's. So you hear about precision medicine quite a lot. This is an enormous problem for Alzheimer's and Parkinson's disease. because the patients we see are incredibly different from each other. Now, you've all heard about this in the context of cancer. For 30 to 40 years, our cancer colleagues have had precision medicines for cancer. Why is that? Firstly, they've been able to sequence the genetic code of cancers, but secondly, they've been able to grow our cancers in the lab. This is a critical point. You can take a little piece of a tumor and you can grow it in a petri dish in the lab. Well, some years ago, the stem cell field learned how to take a small piece of skin or a urine sample or a spot of blood and turn those cells back in time to embryonic stem cell-like cells from an adult, and then turn those cells into brain cells. Those are known as induced pluripotent stem cells. So for the first time in history, we have the ability now to make little brain-like structures from our patients in the lab. So I'm going to tell you a little bit about how those technologies I envisage will transform our ability to match the right drug to the right patient and reduce the number of tombstones that Michael showed for all of those therapies that don't work. The idea here is that some of those therapies may have been perfectly fine, but we didn't know who to give them to. So that's really the key. So here are some of our disclosures. The only ones I'll highlight are actually Rich's, where he is involved with various legal proceedings related to environmental toxicants and Parkinson's disease. And so those are the only ones that are relevant for today's talk. So the precision neurology, I'm going to give you a little vignette. Michael mentioned that we're learning from other fields. There are some patients with Parkinson's-like disorders that come to our clinic and they have a very clear mutation, and those mutations almost certainly lead to a devastating movement disorder. So some years ago, 3 or 4 years ago, I met a young boy who had a devastating disease, very similar to Huntington's disease, but a slightly different gene. And so he was 14 at the time, and what we did is we did for him what I just told you that we can now do. We took a skin biopsy, we made human embryonic-like cells, these iPSCs from him, and we turned those into brain cells. And we used those to test different genetic medicines because we knew what mutation he had. So he has a movement disorder, but he also has seizures. So when we made his brain cells in the dish, we saw something remarkable. You'll see the brain cells of a normal individual on the left-hand side, and you can hardly see them flashing, right? They're just moving very, very slowly, flashing at different times. But on the right side, you see something different. It looks like lightning bolts coming at you. Those are his cells, and they're having seizures in a dish. And so when we designed a genetic medicine for him, we learned how to reverse those seizures in the dish, and we thought we might have a good treatment for him. So 3.5 years, only 3.5 years after meeting him, we worked with Enlorum, with colleagues at Children's Hospital in Tim Yu's group, and we were able to give him a drug. So he's now been treated for 1 year. We wait with bated breath to see what we've done. So far, his seizures are reducing, and he's had the longest seizure-free period of his life just in August. So we don't want to overpromise, but these are the kinds of technologies we can now bring to people with neurodegenerative diseases. Now, Parkinson's is a huge problem. Even beyond Alzheimer's, which of course is more common, Parkinson's is the fastest growing, not neurodegenerative, but fastest growing neurological disease in the world. That's why we're all here talking to you. And precision medicine is a very complicated business for this disease. If you take patients in the Michael J. Fox Foundation, here are about 300 to 400 of them, that do not have a known mutation. These are most of the patients we see in the clinic. What you're seeing here is how they progress over time. If you see lots of squiggle lines that are all over the place, that's what I see too. This is a completely noisy curve, which means that some patients don't progress at all after 5 years, and some patients are severely affected even after 2 or 3 years. What's the difference between these different patients? Are these different diseases, or are they all the same disease? You know, when these patients pass away eventually and give us their brains, we know that there's something very common about them. Michael mentioned a protein alpha-synuclein. That's a protein that we study in our lab. We've studied it for a long time. That is always present in these patients, just like amyloid and tau are present for Alzheimer's. But the way this unfolds for different patients is completely different. And so we seek to study why this is and whether these patients need different types of therapies. And so we fundamentally break this up into 3 different pieces. One, you've heard about this protein synuclein. That's Parkinson's answer to tau and amyloid. Synuclein is this protein that clumps up in the brain of patients with Parkinson's. This protein can fold into a lot of different shapes and can look very different in different people, and we study that intensively in the lab. Secondly, we study genetics. I told you that was one of the recipes for our cancer colleagues figuring out precision medicine for some cancers, so we're very interested in genetics. And third, and Rich is going to talk to you about this in a few minutes, we're really interested in why this is the fastest growing neurological disease. We can only think that it's because of environmental factors and the pesticides and inhaled pollutants that we have unfortunately surrounded ourselves with post-industrialization. So one of the things we can now do remarkably, Michael talked about biomarkers and how revolutionary they are. One of the things we can do is when a patient comes to see us with Parkinson's, we take a few skin biopsies. From one of them, we sequence the genome. From another one, we make their brain cells. But from other biopsies on different parts of the body, we can actually amplify the protein alpha-synuclein. The very same protein that's aggregating in the patient's brain can actually be detected in the skin. And so this is a remarkable innovation because it means that we can not only diagnose our patients earlier, we can tell them what type of Parkinsonism they have, we can also capture that protein in the lab and put it back into their brain cells. So for the first time, we're able to make, like our cancer colleagues, a model that has brain cells and the toxic protein that's causing the issue. So this is quite a revolutionary time for us. It's obviously not just work from our lab, it's depended on many other people, but it means now that companies that work with us can test their drugs in the dish before they go into patients. We can even start to figure out which chemicals bind this toxic protein to make better ways to look at it in the brain. So this is an example of work that we're doing with our colleagues at Mass General and the University College London. And here, we did a screen in human stem cells, and our colleagues have now succeeded in putting the chemicals we found into patients' brains. And this is one of the first visualizations of the alpha-synuclein protein in the brain of a patient with Parkinson's disease. This is a part of the brain called the brainstem. You can see it's very red on the left. That's in the patient where you're seeing the clumping of the protein. On the right, it looks more pale, green and blue. So this is really remarkable. I don't want to overpromise, but it's exciting times. We have 120 more of these molecules to test. This was only the first one we put in patients. So now I'm going to hand it over to Dr. Kralewski, and he'll talk to you a little bit about environmental exposures and how we're using stem cell technologies to better understand them. Rich?
Thank you, Vic, and thank you to the McCourt Foundation for the opportunity to tell you about our work here and to interact with the community. So as Vic very nicely introduced, the environment is very important in Parkinson's disease. And some of the best studied environmental factors that influence Parkinson's disease are pesticides. We know that pesticide exposure at certain levels can increase your risk of Parkinson's disease 2 to 3 times. And in certain situations where there's extensive evidence, such as the case for paraquat, we even recognize that exposure to this herbicide is a cause of Parkinson's disease. And you can see the map of usage of paraquat in the United States from about 10 years ago with the sort of brown and red and yellow areas showing different levels of usage of that herbicide throughout our country. And this is an herbicide that is banned in many other countries. We've been lucky that the public are becoming more aware of this. There's a very nice book by colleagues, Dr. Dorsey and others, called Ending Parkinson's Disease, that speaks about this issue of environmental exposures and what we can do to try to prevent things very eloquently. And then there's also been some high-profile editorials in the New York Times highlighting both pesticide and paraquat exposure, and also exposure to other environmental toxicants like TCE or trichloroethylene as very important for understanding the risk factors in our environment. So we're trying to use the exposure of pesticides and these linkages to Parkinson's disease as a way to understand how a patient's exposure and their genetics influence their risk, influence their progression, and ultimately teach us about the biology of Parkinson's in a way that helps us prevent and treat more effectively. Now, one of the approaches we've taken, if you look at our map there, is for most of the country, we only have very crude estimates of pesticide use. But we're lucky to be working with some colleagues in LA at UCLA who, because of California's pesticide use records going back decades, they're able to actually calculate very precise exposure amounts for patients that they follow closely in the Central Valley, where there's an extensive agricultural industry and there is a lot of pesticide exposure. So using the systems that Dr. Khurana introduced you to, and generating these stem cell-like cells from a patient with Parkinson's disease, we then coaxed those cells from that patient into the dopamine neurons that are one of the neuronal subtypes specifically affected in Parkinson's disease. We generated lots of these dopamine neurons from the patient sample, and then what we did was we used the pesticides that were discovered by our colleagues at UCLA as being associated with Parkinson's disease in their human patients. So we're able to take this data from humans and bring it into the dish where we can do tests and experiments that are impossible to do and should not be done in humans. And so we treated those dopamine neurons in the dish with these pesticides, and then we followed them over time to see which pesticides, when you directly expose them to those patient neurons, cause dysfunction and death. And here's an example of some of the data that we see from that in one of our initial experiments. So the graph on the left, if you look near the top, any of the pesticides that are having dots near the top were not toxic to our neurons. And the pesticides that have those blue dots near the bottom, underneath the yellow bar, were the ones that we considered toxic to our neurons. The list of the pesticides that came up in our screen here are on the far right, and then you see those dark images in panel B here. There's an example of a control well. So this is just a compound that we dissolve different pesticides in. You see healthy neurons all throughout that field of view with their long, what we call neurites, their processes that are stretching out and communicating with each other. And then on the bottom, you see the same experiment, but treated with one of the most toxic pesticides that was in our experiment, propargite, where you see lots of cell loss, and basically you don't see any more of these neurites for communication with cell to cell that neurons use. So aside from being biologically interesting to us, this also has public health relevance in that many of these pesticides are still actively used in agriculture and actively registered with the EPA. A next step that we're able to do in the dish is understand combinations of pesticides. So patients are exposed to different levels and different combinations of pesticides, especially those that are out in the fields working. And so we looked at a specific set of pesticides that are applied in cotton agriculture. This so-called cotton cluster, we were able to take a set of those pesticides And in the bottom left, you see that tiny graph is a recombining of all the different possible combinations. So we could ask, are there some combinations that are actually more dangerous or more problematic? And are there some combinations that are less problematic? And what I've done there on the right is sort of just blow up a little part of the graph to show you that the sort of lower the bar goes, the more toxic or the more detrimental that pesticide combination is to the cells. And so we saw that a combination such as trifluoralin and tribufos had more cell death than either of them by themselves, which suggested to us that there might be some combinations that are particularly dangerous and need to be thought about carefully by regulators. So we want to leverage what we've learned from these experiments. They can help us identify some of the most dangerous pesticides. They allow us to test for combinations of pesticides in controlled ways that is not easy or possible in other systems. And then we can do the important biology on those cells. What types of pathways are being influenced by these pesticides? How can we potentially add protection to those vulnerable pathways? And then we also want to help provide additional evidence for regulators who are making decisions about whether to allow these pesticides to be applied and how to regulate them. And then getting to our interest in genetics, all of the experiments and data that I showed you on the last few slides were really with just a couple different cell lines from specific patients that had a genetic mutation that contributed to their Parkinson's disease and caused their Parkinson's disease. But most patients that come into the clinic have much different genetics. They don't have these mutations that will unequivocally cause Parkinson's disease. So we want to understand how are other mutations making them more or less vulnerable to what they encounter in the environment. So we're able to now, with advanced computational technologies, pool many different cells from many different patients into one dish and experiment that allows us to expose them to the exact same conditions and see how do those different genetics from those patients result in their responding differently to the same pesticide or the same stressor. And so we are excited about those experiments. Another more recent collaboration that we're working with colleagues from UCLA again, and then colleagues at Brigham and Women's, Dr. Koretska-Roth and Dr. Cox, who are experts in studying how the microbiome seems to influence cognition in Parkinson's disease models. So we know that different types of bugs from the gut of Parkinson's patients can influence outcomes when the mice are exposed to those different bugs. And so we're trying to then use our different cell models to understand how pesticides might influence that microbiome as a possible way to affect cognition in Parkinson's disease. So with that, I'll pass it back over to my colleague.
Thanks, Rich. So I'm just going to wrap up now, just a couple more comments. But the idea is, what we tried to convey is that this is a heterogeneous disease. There are genetic factors, there are environmental factors, and we're trying as a group to figure these things out. But we also need a mechanism, because humans are complex, to be able to try drugs that are considered safe by the FDA and actually observe very carefully for how our different patients respond to them. So I run something called the Harvard Biomarker Study. Michael and others helped very much many years ago set this study up, and I've inherited it in the last couple of years. It's now called HBS 2.0. And within this, there's a program called MyTrial. And I'm just going to tell you a little bit about what this is. Basically, with MyTrial, we take small groups of patients that see us at the Brigham and at Mass General, and we bring them to our clinic and we deeply quantitate their biology. We put accelerometers on them, we sequence their genome, we make stem cells from them. We start to do PET scans in their brain to start to analyze their biology. And 6 or 12 months after we do that, we actually give the patients a drug, and we give their stem cells a drug at the same time. And we try and figure out if all patients respond equally to a drug or patients have very different responses. So for example, Michael talked about an Ozempic-like drug called exenatide, a GLP agonist. Sometimes it seems to succeed for Parkinson's, sometimes not. The question is it a real drug for some patients, or is it just not a drug? Is it a flop? So we're trying to figure that out. We're collaborating with Sanofi to bring that drug into our MyTrial program and definitively answer that question. So this is really just a schematic of this. We take a population, we track patients over time, we make stem cells, and then we do a PET scan to look at something related to how that drug works, and we try and figure out if the patients respond or they don't respond, and we make stem cells from them and we ask exactly the same question. And what we're trying to do is say, we're not going to put patients in a big clinical trial that don't have a great chance of responding to this drug. Otherwise, we're wasting their time and we're wasting the drug. We're not matching these drugs appropriately to the patients. So this is really the idea, to create an optimized clinical trial. This is an example of this working for a very aggressive cousin of Parkinson's disease called MSA, multiple system atrophy, same protein synuclein, devastating disease. Here you can see a patient whose inflammation is going up in the brain over time. She's given the drug, she responds, but many other patients in this study did not respond to this drug. When we made the brain cells of these patients in the dish, we could actually predict perfectly so far who in life responded or didn't respond to the drug. So this drug has now failed clinical trial and it's been put into the graveyard, to use Michael's metaphor, but it's possible that this drug was actually good for about 30 or 40% of the patients who are in this trial. So we continue to work on projects like this to try and make precision medicine a reality. This is really the conclusions. We think that this is the time for thinking about precision medicine, coupling these ideas to what you heard from Michael. The earlier, the better. There's no question about that. And two of the innovations we've talked about today are stem cells. We talked about that in terms of the genetics and the environment, and also these really important emerging biomarkers. So I'm going to leave it there. We have a lot of people to thank, but I'm just going to thank the most important, our patients and their caregivers, because they're the ones that make all of this research possible. Thank you so much. We'd be happy to take your questions.
From this intimate group who's here, if there are any questions, happy to start with any you may have. Miguel is sporting a mic. If you think of some, please raise your hand or tap Mike on the shoulder. But there were a few that came in online, at least in advance, and maybe there's a mechanism for anyone online now. But having seen the ones that have come in, maybe I'll turn to Vic with a couple of questions that are sort of thematically linked to this question of whether we should be trying to find just the unique needle in the haystack, precision medicine, one treatment for one patient, versus trying to find a universal therapy. So there's two questions that relate to that. One is, can the breakthrough cure for Huntington's disease, there's a recent advance that looks hopeful, genetically based for Huntington's disease. Can that also help with Parkinson's disease and dementia and Alzheimer's and the like?
Okay, so I'll take that. Some of you will have, it got a lot of press recently after many failures. A genetic approach for Huntington's disease has shown some value. We are still awaiting the actual publication and the details of that, but what was released looks very, very promising. Huntington's disease is one of those diseases that Michael mentioned in his talk as having a very well-defined genetic cause, and so it was it's a great candidate for a gene therapy. As I just said, Parkinson's is much more complex, but there's a couple of things that give us hope from this. One is the protein alpha-synuclein that also can aggregate and cause neurodegeneration in Parkinson's is now the subject of multiple gene therapies and antibody-related therapies, and so the Huntington's work gives us hope that when you design a clinical trial correctly and the therapy doesn't have its own toxicity, which may have been a problem for earlier Huntington's trials, then there's a chance that we can really shift the needle. I think, though, that what Michael said about the earlier the better is critically important for a protein like alpha-synuclein, and so we'll just have to wait and see. But that is very much a possibility. The second way in which this could be very important is that we and others are looking at genetic targets that actually encompass, speaking to Michael's question, 20 to 30% of patients with Parkinson's as opposed to a very small number. If we can turn the complex Parkinson's disease landscape into 3 or 4 subtypes, covering 20 to 30% of patients, then this kind of result with Huntington's disease makes us hopeful that we can define and develop genetic medicines for large groups of Parkinson's patients.
And then specifically, the treatment that they're finding for Huntington's, will that be helpful for Parkinson's?
The very same treatment we do not anticipate will be helpful for Parkinson's because that is very specifically targeting the protein that is causing Huntington's disease. So unless there's some new science here that we're not aware of, we don't expect that to be a treatment, but we expect the general approach to be very applicable to Parkinson's and worth trying.
Thank you. Related to that, perhaps you've already addressed this, but this is a broader question. Do you believe that discovering a cure for one neurological disease or neurodegenerative disease can pave the way for cures for more effective, for others?
Yeah, this is a really interesting question. So one of the things I'm saying is, wow, this is really complex. We may need lots of treatments. Now, Michael's saying, what about killing many birds with one stone? Is that possible? So the one thing that we're thinking about very carefully, Brian McCourt mentioned it a little bit earlier, was nasal furalimab, which was an anti-CD3 monoclonal antibody that was originally developed for multiple sclerosis. It is now being in trial for Alzheimer's disease. Why is it being tried for multiple diseases? Because brain inflammation may be driving progression in multiple neurodegenerative diseases. And so this type of treatment that dampens down brain inflammation may actually be an example of a treatment that could be useful for more than one disease. We have recently discovered, with the same type of PET scan I just showed you, that brain inflammation happens in that disease MSA. It also happens in Parkinson's. So we have now started our clinical trial for that drug in MSA, and next year we're hoping to start that for Parkinson's.
Thank you.
A question over here. Is there one approach to multiple diseases? So Parkinson's and Alzheimer's, the number of patients that get it goes up as they age. So there are research groups out there that are trying to target aging mechanisms, and we think that there's a reasonable chance that if we're able to alter some of those mechanisms that cause the body and the cells to age, they might help at least slow. We don't necessarily think it's going to stop or reverse, but might slow the process that makes older patients vulnerable to Parkinson's and Alzheimer's and some of these age-related disorders.
So some strategies may be good across the boards and others may be very targeted, not only specific diseases, but specific subtypes within a disease. Miguel, did you?
Sorry, we got here a little bit late. The traffic coming in from Maine was brutal today. But I had a question related to this sort of research, recent, I guess, success at Santa Fe. Was that because they were able to effectively penetrate the blood barrier and be able to get treatment more effectively into the brain?
Yeah. I think you'll have to clarify a little bit, but if you're thinking about, so Sanofi and Roche both have antibodies to alpha-synuclein, if it's their Parkinson's program that you're alluding to. And these new antibodies have what are called brain shuttles attached to them, and they cross the blood-brain barrier more effectively. So I think you're referring to that. And I think that this is a really exciting advance, and there's a lot more work to be done, but getting antibodies into into the brain is one thing that is going to be important. There's a possibility, we're actually testing this with Sanofi right now. There's a possibility that their shuttle may even get the antibody into brain cells better, which would be great for a target like alpha-synuclein, but we don't know that yet. But yes, these delivery methods are really important. The company that's furthest along with this kind of technology that I'm aware of is Roche, that is pushing forward a program that Michael knows very well, and he mentioned it as well, related to prasinezumab, one of their antibodies for Parkinson's.
Well, the other thing, a comment I'd like to make as a retired epidemiologist, the focus on the environment is just so profound. And one of the things anecdotally that struck me was I had occasion about 10 years ago to attend a high school reunion with my wife. And in the course of meeting a number of her classmates, it turned out that quite a few of them had come down with some form of cancer. Now, this took on added significance to me because the reunion was held in Niagara Falls, in a town that actually abuts Niagara Falls, where there is a preponderance of major chemical companies. And in fact, my wife's father, who was a PhD chemist and ran one of the largest labs, was always talking about the implications of this kind of exposure. So I can only wonder whether my wife, who has now been diagnosed with Parkinson's, may also, growing up, have been exposed to many of those same pesticides that they make there.
Right.
Just a comment.
No, and thank you for the comment and your insight. What we can do now, what our hope is, and this has really been work that we've also learned a lot from Dr. Schwarzschild on his early work with environmental factors. What we are hoping with our stem cell technologies, and this work I'm collaborating with Rich on, is that we can move beyond speculation. We can actually grow someone's brain cells, expose them to pesticides, and actually ask the question, were they particularly sensitized to that because of something in their genome, and what's the mechanism of that? So we hope that we'll be able to answer these questions more effectively one day.
Thanks. And another question here.
Hello, thank you. I had a quick question. So how many levels of Parkinson's is there?
You mean like clinically? It's progressive disorder, so there are many different staging systems.
Yeah, stages.
Yeah, so you can have a scale from 1 to 200, and then every point is an increment, or there are scales that go from 1 to 5 in Parkinson's disease. So it's sort of how you slice it. It's kind of arbitrary because Parkinson's, unlike strokes, doesn't generally take a step forward and you get to the next stage. It's a very progressive, as anyone who's living with it generally knows. So it defies an easy way of saying there's 3 stages or 10 stages, but there are, it does progress. So that can be very helpful to do that, to slice it up that way.
Okay, that makes more sense. So what are the best habits to have to keep like the Parkinson at like the current level you might be at? To not, I mean, I know you talked about prevention at every stage. So what are like the best habits to kind of maintain that stage? Or is that impossible? It's always going to be progressive?
Nothing's impossible. People and things vary as those spaghetti lines that Vic showed for individual journeys taking their own path. And some people will seem to have minimal progression, and some will progress far too rapidly. What it is that distinguishes those people, whether it's genetic, environmental, like, is a big mission for research. If you look at those, the vast majority, even the ones that had the best progression over years, still tended to have a progression. So the realistic aspiration is what we can do to keep it from progressing rapidly. There, we have very few clues. There are a lot of leads that haven't been substantiated. As you may know, probably the best advice, and it's based not just on hopefulness, but actually on real data, is that exercise, regular exercise, use it or lose it, as simple as that is, can make a big difference. So without necessarily entering triathlons, being active with one really enjoys and sticking with it is one of the most likely things you can do to make a big difference.
Awesome, thank you.
Sure. Related to that, maybe Rich, and the thoughtful question about environmental factors. Another question that was asked was another association from the environment, if you will, which is head trauma, and whether that has an impact on the disease. And
Yeah, so we do know that head trauma is a risk for development of Parkinson's disease, and there's some other really interesting data from the pesticide epidemiologic literature in that there's even some specific pesticides that the combination of some head trauma and exposure to that pesticide seems to sort of skyrocket your risk for Parkinson's disease. And so that we assume, and we hypothesize that that is because those certain pesticides are working on some part of the biology in a way that's more affected by that additional sort of second hit of the head trauma. And so yes, we do know that trauma, that head trauma specifically, is a risk factor for Parkinson's disease.
do you have any questions for the audience, or are there other questions? I have one, just as it comes to mind, with the modeling and this innovative approach that can be high throughput by putting a model of one's brain with Parkinson's in addition and exposing it. You mentioned head trauma and the interaction and the like. So some things, it seems, are going to be hard to model in a dish. Head trauma interacting with chemicals, although different chemicals. Have you been able to validate the interactions and like either with finding that the two that you found that were conspired to create more damage in the dish, was there epidemiological evidence that those two in combination are associated with more difficulty?
Right, so the reason we did that experiment is because as you start to ask more specific questions about just these two pesticides, what happens is the studies aren't big enough and we don't have enough data to be able to ask these single pairwise comparison type questions. But we do know for other pesticides, there has been evidence of sort of conspiring together. So paraquat and manab are an example where the individual risk of each one by itself, when combined, seems to be more extensive. And so what we're trying to do in the lab is better understand if we put that pesticide on the cells and we put the other pesticide on the cells, what is the cell's response to pesticide 1? What is the cell's response to pesticide 2? And then once we understand that biology better, then I think we might be able to go back to our epidemiologic colleagues and say, are there any patients that have a mutation in that pathway and this pathway, and can we take a closer look at them? But in many ways, we're limited by the quality of data, and that's why we've been lucky to work with our colleagues at UCLA who have this very nice exposure data that even opens up the possibility to do these studies. But
I mean, I was just going to draw attention to something you showed as a way of just clarifying Michael's, an answer to Michael's question, and that is that the original epidemiologic data, for example, for cotton agriculture, suggested that certain combinations of pesticides were synergizing to create Parkinson's, and when that entire set was screened by Rich, he found the same combinations would be synergistically toxic in the dish. So in that original screen, there was a very nice parallel between what was found epidemiologically and what was found in the dish. And that's how we identified a particular pesticide called trifluralin that he's actively studying very much right now, because it seems to be a hub. And so if you just removed it, it actually could be that you can use a whole bunch of other pesticides safely, but when they're used in combination with this, it could make them a lot worse. So if we can find the mechanism of this, which we're all trying to do right now, that could be a very important finding.
Thank you. If there are no other questions, it is a lovely day. Oh, yes, please. Here's the microphone just so others can hear too. Thank you. There you go.
When you're saying pesticides, do you mean like, keeping our landscaping like a nice, so that it's looks nice and the bugs don't, like eat up our garden? Or, I mean, or what other kind of pesticides are you talking about? Because I don't know, pesticide A and B, if they get combined, they're deadly to the brain.
Right.
Parkinson's, is that?
Yeah, so some of the pesticides are restricted use in that you can only apply them if you go and get training and have a license. Some of them you can buy at
We're talking about gardening.
Yeah,
like
yeah.
landscaping.
Yeah.
Not other stuff.
Right. Some of them for your gardening and
Or like we're
landscape.
cleaning, like for example, we're cleaning
Yeah.
our house and stuff.
Right. So some of the pesticides that are used commercially are the same ones you can buy at Home Depot for your landscaping. And so that's why we want to make awareness of which of these pesticides seem to be most problematic. Because if they are available off the shelf, we would suggest that maybe that's not the best idea to have them available off the shelf, and understanding that if in your garden you're using pesticide A and B, you should not use those two together. And this is the data we're trying to build on here. I mean, and it's not just commercial farming, right? Go ahead.
It's a good thing I don't have a nice garden, clearly.
Well, people living closer to golf courses, higher risk of Parkinson's disease. There's very interesting studies on this. I don't know if Michael knows more about those, but they're interesting observations about these. I'm taking a...
That might be an indication, sorry, I won't...
Of the pesticide use that happens at the golf courses use a tremendous amount of pesticides and herbicides to control weeds on the fairways.
You're explaining a lot more than I was expecting to.
Just one simple take-home that anyone can agree on in this domain is that there's labels on the back of all the home pesticides that are rarely followed, like using gloves and masks. So at a minimum, I'm not sure if there's more specific advice, but that seems a level of caution that is warranted by the data. But other than that, I don't know if...
Well, I guess I'm going to bring this back a little bit to a public health message and a message about lobbying and talking to your local, representatives, political representatives. Many of the chemicals that Rich is finding, these are banned and have been banned for many years in other countries. And they've been banned on the basis of NIH research. So when people ask us, why is it that NIH research is not converted into better outcomes for Americans, I sometimes think NIH research is translated to better outcomes for people overseas, because NIH research has really highlighted the problems with some of these chemicals, but they are not banned in this country. So it's just worth thinking about and being mindful of. Paraquat, paraquat was banned decades ago in Australia and in Europe, and it should be thought about much more carefully in the United States as well. So I think there's a public health message here as well, and it really feeds into medical research and so forth.
And it needs to be a nuanced dialogue with, and actually paying attention to the data closely for different pesticides, because we need to feed the population. Like, we need to make sure that our agricultural communities have the tools they need to produce and keep everyone fed. So we're not saying every pesticide ever should be banned, but we just need to think very critically about it and follow the evidence and develop more evidence.
Well, and the individuals using it should be careful on how they use it so that they don't harm themselves. Because if you're harming yourself, I mean, it's unproductive mostly. But then the other part of the prevention part, that was very intriguing because I never thought that Parkinson's was preventable. I thought that that was like a disease that came on from head injury and just because of the people we see affected, the famous people we see. Like, okay, now Alan Alda has it, but we don't know how did he get impacted by Parkinson's.
Well, we won't
Right?
figure it out unless we
No.
study it, and that is how we will get there. I think things that we have made, public health approaches to prevention, even if there's political slippage. There's fluoridation in the drinking water as an example, that where tooth decay is reduced. These are things where the science is followed, and it can lead to it. So I think while particularly Parkinson's is very complex, has been highlighted, and involves genetic and environmental influences that may be very different in different people. It's only by understanding how that works that we will be able to make it less nihilistic and make it worth pursuing. Yeah. Well, thanks for your thoughtfulness, and I think while small, the audience here from your epidemiological and public health perspectives reflects the inquisitiveness and the searching for answers that will get us there, whether it's through the labs, through public policy. So I applaud your joining in this enterprise. And if not, I'll thank the McCourt Foundation and once again, and you for joining in today, and those listening in for your interest in trying to make some progress. So thank you, my colleagues as well.